Taking a look at another biological molecule, the lipids. Lipids are a very diverse group of chemicals. They are all organic molecules which are insoluble in water. Most lipids are formed by fatty acids combined with an alcohol. The most common form of a lipid is a triglyceride. Your lipids are usually known as fats and oils. Fats are solid at room temperature, and oils are going to be liquid at room temperature. However, chemically, they are both very similar. Fatty acids are a series of acids, some of which are found in lipids. They contain the acidic group, the carboxyl group, that's your carbon, oxygen, oxygen, and hydrogen. The carboxyl group forms the head of the fatty acid molecule. Common fatty acids have long hydrocarbon tails attached to the carboxyl group. So when you look at your diagram, you have your long chain of carbons. They are saturated if all of the bonds between these carbons are single bonded. If you have double or triple bonds between the carbons, then you have an unsaturated fatty acid. At the top, you have that acid head. As you're looking at this, you can see your carbon, your hydroxyl group, and then your double bond oxygen. The tails of some fatty acids have double bonds between the carbons. Again, those are going to represent your unsaturated fatty acids because they do not contain the maximum possible amount of hydrogen. The double bond makes them easier to melt. If there is more than one double bond, the fatty acid or lipid is considered a polyunsaturated. If there is only one double bond, then it's considered a monounsaturated. Animal lipids are often saturated and occur as fats. Plant lipids are often unsaturated and occur as oils, such as olive oil. This is just showing you when you have your carbon chain. If there's only one double bond, that's considered a monounsaturated fat. But if there is more than that, here you have one double bond and then a second double bond, that's considered a polyunsaturated fat. Fatty acids that contain all single carbon bonds, the maximum number of hydrogens on each tail, this is going to represent your saturated fatty acids. They occur as solids at room temperature. Notice there are no double bonds. These are all single bonded carbons. Therefore, this is a saturated fatty acid. When we look again at the unsaturated, there's a double bond, there's a double bond, and another double bond between your carbon atoms. Makes it unsaturated. Since there's more than one, that would be a polyunsaturated. Taking a look at alcohols and esters. Alcohols are a series of organic molecules which contain the hydroxyl group, your OH group, attached to a carbon atom. Glycerol is an alcohol with 3 hydroxyl groups. This is a glycerol molecule in this green box, and you'll notice it contains 3 hydroxyl groups. The reaction between an acid and an alcohol produces an ester. The chemical link between the acid and the alcohol is called an ester bond or an ester linkage. Here's the acid, that carbon, oxygen, oxygen, hydrogen. Combined with an alcohol, there's your hydroxyl group. It's just been flipped. Remember, they need to line up when they bond together. After the reaction occurs, you're going to have an ester. And then In order for these two to combine, that acid and alcohol, for them to combine together, water is going to be produced. If water is produced, that's a condensation reaction. This bond can be broken by putting water back in, reversing the process. Remember, that's hydrolysis. Your COOH group on the acid is going to react again with that hydroxyl group on the alcohol to form the ester bond. And here's where our ester bond would be formed. Again, water is released because it's a condensation reaction. Triglycerides are made by the combination of 3 fatty acids, hence the name triglycerides. Tri means 3, so 3 fatty acids, with 1 glycerol molecule. Fatty acids, they're organic molecules, again, that contain that COOH group attached to your hydrocarbon tail. The glycerol is a type of alcohol. If you'll remember from 9th grade biology, triglycerides represent kind of like that capital E shape where the backbone was the glycerol, and then your 3 fatty acids. Looking at this structure, here's your glycerol, and then the 3 fatty acid chains. It's just another representation where you have that backbone as glycerol and then the 3 fatty acid chains. The tails differ in length depending on the specific type of fatty acid that's involved. Each fatty acid molecule joins to a glycerol by a condensation reaction. When a fatty acid combines with glycerol, it a glyceride. So here's your glycerol, your 3 hydroxyls for your alcohol, and then your 3 fatty acids. Each one of those is going to contain an acid on the end, your acid functional group. And then part of the acid, that OH on the acid, and then the hydrogen from the alcohol, they're going to form water molecule, hence why it's called a condensation reaction. So this gives us a total of 3 water molecules. Glyceride is an ester formed by a fatty acid combining with the alcohol glycerol, and when they're bonded together, you're going to have that ester bond. It's just another diagram showing you've got your glycerol with the alcohol, that OH group, and then your 3 fatty acids, each one of the fatty acids showing you the acid functional group. They'll go through condensation reaction where water is produced. Again, there's going to be 3 water molecules produced. When they bond together, you're going to have an ester bond forming. So glycerol, the 3 fatty acids go through condensation reaction, 3 water molecules, and then you have the ester bonds attaching your fatty acid to the glycerol. Triglycerides are insoluble in water, but soluble in some nonpolar organic solvents, such as chloroform, ethanol, or ether. Fatty acid tails are nonpolar, so they do not mix in the polar solvents like water. They are therefore said to be hydrophobic. They do not mix freely with water molecules. Looking at the functions of triglycerides, lipids make an excellent energy reserve because they are even richer in their carbon-hydrogen bonds than the carbohydrates. A given mass of triglyceride will therefore yield more energy on oxidation than the same mass of carbohydrate. Basically meaning lipids have a higher caloric value, which is an important point for storage products. Animals that hibernate store excess lipids for energy. Fat is stored throughout the human body, just below the dermis of the skin and around the kidneys. Below the skin acts as an insulator against the loss of heat. In your marine organisms, this is called blubber. It's a triglyceride that helps with insulation, such as what's found in whales. It's also going to aid in the buoyancy of the organisms. Lipids can also act as a metabolic source of water. When oxidized in respiration, they are converted to carbon dioxide and water. And this is important in dry habitats. This little guy is known as a desert kangaroo rat, never drinks water. It survives on the metabolic water from triglycerides contained in the foods that it eats. Phospholipids are a special type of lipid because one end is soluble in water. Because one of the 3 fatty acid molecules is replaced with a phosphate group, which is polar, therefore it dissolves in water. Remember, like dissolves like. The head of the phospholipid is the polar region, it's hydrophilic. And then the tails, these are the tails, are the nonpolar and they're hydrophobic. The phosphate group is hydrophilic and it makes the head of the phospholipid molecule hydrophilic, although the 2 remaining tails are still hydrophobic. This allows phospholipids to form a membrane around the cell. Hydrophilic heads in the water solutions on either side of the membrane and their hydrophobic tails forming a layer that is impermeable to hydrophilic substances. Okay, you need to be familiar with the testing for lipids. Lipids are insoluble in water, but soluble in ethanol, which is a type of alcohol. This allows for an emulsion test for lipids. The substance that's going to be tested is shaken vigorously with some ethanol. This allows any lipids in the substance to dissolve in the ethanol. The ethanol is then poured into a tube containing water. If lipids are present, a cloudy white suspension is formed. And the cloudy white is your key words that you're looking for when you write down your descriptions if a lipid is present. If no lipid is present, the ethanol mixes with the water. Light can pass straight through the mixture, so it looks transparent. If lipid is dissolved in the ethanol, the mixture is considered an emulsion. And an emulsion is going to give us that cloudy white suspension.